So without further ado, we have a lovely speaker with us today. This is Dr. Gutenkunst. Some of you, if you're in classes or being in NCB. So Dr. Gutenkunst did his PhD at Cornell in physics. And then he completed 2 postdocs, one at Cornell and then one at Los Alamos National Laboratory. And he is now an associate professor and also our associate department head in the Department of Molecular and Cellular Biology. And he's one of our long-standing mentors. His research is quite interdisciplinary, I would say. His lab aims to understand, the evolution and function of the complex molecular networks that underlie life. And he does that by integrating many different things, including computational population genomics, bioinformatics, systems biology, and molecular evolution. And as a random fun fact, Dr. Gutenkunst is also a triathlete. He finished Ironman Arizona last year in just under 14 hours, right?
Yes.
Yes. So for those of you who don't know anything about triathlons, that's a 2.4 mile swim, 112 miles of biking, and a full marathon under 14 hours. So he's not going to talk about running the area today, but as you can tell, we're going to be talking about inferring natural selection from population genetic data. So thank you for being here today.
Thank you, Gavin. Quite the introduction. You actually stole my first couple of slides. So I'll reiterate a few things. I'm just going to ask you to talk a little bit about my journey, about how I got here. So I actually did my bachelor's in physics. So I grew up. So for me, high school biology was color the mitochondria purple, color the Golgi apparatus blue, and it was mind-blowing. It was just pointless memorization, and I hated it. So I studiously avoided biology as an undergraduate. I took the one required class and put as little effort into it as I possibly could. And so I went to get my PhD in physics and thought I would work on fluid mechanics. I took my first class in turbulence and learned that nothing fundamental had been learned in the last 50 years. And said that kind of sucks. But I was in this environment where I was being exposed to lots of biologists started going to research talks. It realized, hey, it's not all stamp collecting. It's not all memorizing these big complicated pathways, but there really are some key critical ideas here. And what became really exciting to me is that I was coming from physics where it felt like there were a few big problems, and to make progress, I would have to be smarter than the 150 other people who were working on that problem. Whereas in biology, there are so many problems. There are so many things we can work on and think about. And oftentimes, the art is not in solving the problem, but in finding the correct problem to think about. And so I was in a physics department. I was sort of trained as a physicist, but I was really doing computational systems biology. So trying to understand how signaling networks within cells worked. And the more I thought about these networks, the more I realized that, as a purely secular perspective, it's like a miracle that all this stuff works. Right? It's just amazing that a cell actually works and generates some functionality, and that 1 trillion cells work together to make you. Right? It just boggles my mind. And so I got really interested in what is the process that led to these systems assembling that can do these amazing things. And that really led me to thinking about evolutionary questions. And so I did a short postdoc in population genetics, basically because that was the field where I could find a mentor who was asking some of these questions and where I could use the skills I had. And as Jen mentioned, I did another short postdoc at Los Alamos. The national labs are really cool places to do science, really weird places to do science, but lots of cool ideas there. And then I came to Arizona about 12 years ago. Turns out I'm a joiner, so I'm associated with programs of applied biosciences, applied math, cancer, genetics, statistics, evolutionary biology, biostatistics. So one of the great things about the University of Arizona for me has been the ability to make these connections, to draw students from all across campus. I've had 4 PhD students. None of them have come from my home department. I think it's really important that you as students and me as mentors feel empowered to go anywhere you can, right? You have the ability to make impacts on all kinds of scientific problems, and you don't need to feel restricted by the nameplate that says, oh, this person's doing cancer biology and this person's doing something different and they can't talk to each other. Really, the best ideas come from those sorts of connections. And as Jen mentioned, I've had a whole slew of UBRP students come to my lab. I have 2 right now. And I won't call them out and embarrass them yet. So outside of the university, I have kids, they're awesome. My wife, she's awesome. As Jen mentioned, I'm a little bit obsessed with triathlon and skiing. Okay, so the scientific field I work on is population genetics. And so I've structured today to not so much be about what my lab does, but to be sort of an introduction to the field and some of the ideas. And we'll see whether we get to what my lab does. If we don't, that's perfectly fine. So population genetics as a field is really about understanding the genetic differences that exist between individuals within the human species, within any species, but understanding how those mutations and those genetic differences are created, how they spread, and how they affect the biology of the organism. And I think of population genetics as in sort of the micro-theory that underlies evolution. That we can think about evolution over long time scales, that dinosaurs, those that survived the meteorite, eventually evolved into birds, and our common ancestor evolved into chimps and humans. Population genetics is sort of about the small little changes that go in and add up to make those big changes to create this diversity of life we see on Earth. And let me just say, please stop me at any time to ask questions. I'll be really, really upset and disappointed if you don't ask questions. I'll be offended. So from a practical perspective, one use of population genetics is that it's the foundation of genome-wide association studies. Raise your hand if you know what a GWAS is. Okay, 3 or 4 people, that's about right. So if you read in New York Times that they found the gene for loving coffee, how does that happen? Anyone know? Any guesses? Okay.
A lot of people and compare surveys and just try narrow it down.
Yeah, exactly. So a genome-wide association study is simply we have some phenotype, some characteristic that we can measure about people. It might be their height, it might be their whether or not they got a certain disease, it might be how much they love coffee. And so we have that information about a bunch of people, and then we also have information about their genotype. So across their genome, what mutations do they carry? And a genome-wide association study is simply asking, is there a relationship there? So we can go through the genome and say, at this particular position, 20% of people have a T, 80% of people have an A. Does having a T make you more likely to drink coffee or not? So we can ask that all across the genome. And if we do that, we'll get some sort of plot like this. So this is what's called a Manhattan plot. And so what's shown here are all 23 human chromosomes, where in each of these dots is a different mutation that they tested. And in this case, this is a study from a couple years ago looking for genetic associations between a person's genetics and whether or not when they got COVID, they had a very severe infection that led to hospitalization. And what's plotted on the y-axis here is a p-value, so a measure of how strong is the relationship between the genetics and application and the phenotype here, which is severe COVID. And what you see is there's a bunch of noise, and then this line is sort of the cutoff. And there's a whole cluster of genes here that bounce across the cutoff. And this is a signal here that one of these genes maybe has something to do with COVID response, or more specifically, genetic variants, mutations in those genes have something to do with COVID response. And so, this paper would be accompanied by a story in the New York Times about, we found a gene for long COVID, or severe COVID. But of course, there's a lot of work that needs to be done from going from a list of genes that have something to do with COVID to actually understanding what their biological effect is. And so the whole basis of this sort of study is population genetics, the field like this. Population genetics has also given us recent, really remarkable insights into human evolution. So it turns out that this little piece of DNA that some people carry that makes them more susceptible to a severe response when they get infected with COVID is actually something that was inherited from Neanderthals. People know this story? How much of your DNA is Neanderthal? So if you have European or Asian ancestry, it's probably around 3%. So when humans left Africa, the genetics says at some point we interbred with Neanderthals, and some of that DNA is still sort of circulating around in our population. Pretty cool story. And most of it, probably doesn't do very much in most of our lives, but some of those little bits of DNA influence the phenotypes. And then it turns out there's a lot that involve various immune responses. So really cool biology. So the first concept I want to expose you to from population genetics is this idea of genetic drift. And I'm going to use a bunch of words here. So allele frequency. So here, allele, you can think of as just whether or not you have a mutation. So they're going to change over time stochastically. So what that means is there's a lot of randomness involved in the process. One sort of lesson you need to take away from anything from this talk is that evolution is really stochastic. There's a lot of randomness involved. And so if we were to take, for example, some mutation that, say, take a whole bunch of different mutations that all started at 20% in the population. So 20% of people carry them. If we were to follow those over time, the frequency of those would bounce around. Sometimes they would get more common because the individuals who carry that mutation just happen to have more offspring than individuals who didn't. Sometimes the mutation would become rare because it got unlucky, and the individuals who carried it happened to have fewer offspring. And that frequency, the frequency of that mutation would bounce around from generation to generation. And it turns out that drift, being driven by randomness, matters more if the population is small than if the population is big. So here I'm comparing a simulation for a population size 100 to a simulation of a population size 1,000, and you'll notice how much more quickly the allele frequencies change in this top graph versus the second graph. And so this really matters in contexts where we have population sizes getting small. Like when humans left Africa, it turns out it was a relatively small population of humans that went through a bottleneck, and their descendants then sort of settled the rest of the world. And so going through that bottleneck sort of accelerated the drift in that population. This also happens in the context of sort of endangered animals. Do you have a question?
What does one line represent? Is it like a family or?
So one line, good question. So this represents, so if we were to go out and measure how common this particular mutation was, over time, through a bunch of different surveys. So if at this time t equals 0, we sequenced everyone in this room and said 20% of you carry this mutation. And then we went on 100 generations from now and sequenced the students in UBRP at that point, and maybe we would find that 50% of them carry this particular mutation. Does that answer your question?
Yeah.
I hope UBRP is not a pan-mictic population. So, genetic drift is all about neutrality. So what happens when mutations don't do anything? But the most interesting mutations, of course, are those that change the phenotype, that affect fitness. And so that brings me the idea of natural selection. And so some of the way we think about this mathematically is the idea of a selection coefficient. That a mutation that has a selection coefficient S changes the relative fitness of an individual to 1 plus S. So if we have a selection coefficient of plus 0.1, that means that an individual who carries that mutation is 10% fitter. That on average, over time, they'll have 10% more offspring than someone who doesn't carry that mutation. Or S could be minus 10%, which means that mutation harms fitness. And individuals who carry that mutation will tend to have fewer offspring. And mutations can be either deleterious, right? They can be bad, they can reduce fitness. Neutral, not have much effect on fitness at all. Or advantageous, right? There's some mutations are good for us, right? That is the grist of evolution that enables populations to adapt to an environment. And so, if a mutation is advantageous, has a positive selection coefficient, then over time its frequency will tend to increase. Right? It is favored by natural selection. So it will tend to go up in frequency, but evolution is stochastic. Randomness matters. And if a mutation is deleterious, it will tend to decrease in frequency over time and get purged from the population. Again, that is the action of natural selection. Any questions?
Actually, I have a random question. What do you use to make your figures? Like what program do you use?
This, I think, is not fun. Okay, another piece of vocabulary is the idea of fixation. So the mutation fixes if it takes over the population. So if we started out at 20% of the UBRP population carried a particular mutation, and we came back 100 years of UBRP later, and everyone carried that mutation, we would say that mutation is fixed. And this is how you get differences between species, or mutations fixing in one species versus the other. And this process is really heavily studied in population genetics, and there's some cool mathematical results that I think have important implications for how we think about how evolution works. So the first is that for new beneficial mutations, the population size doesn't matter much. So what I'm plotting here is probability of fixation for mutations that are slightly good, 1% fitness advantage, or slightly bad, minus 1% fitness deficit. And these lines show different population sizes. And so we start with a relatively small population of 100, and then we have 1,000, and then hiding behind that 1,000 is the n equals 10,000. And what I hope you can see is that for the beneficial mutations, as we go to the right, all these lines are the same. And it doesn't matter really whether you're in a population of 10,000 or a population of 100. If your selection coefficient is 0.01, you have about a 2% chance of eventually taking over. On the other hand, if we consider deleterious mutations, so here I'm plotting the same graph now for mutation, but now I'm sort of zooming in on this part by changing the y-axis. And so if we focus on the deleterious mutations here, this is the probability that a deleterious mutation, something that reduces fitness, takes over the population. And you can see that if you look at the scale, it's on the log scale. So here's a probability of 10 to the minus 18, right? That is like a 1 in a billion chance. that a mutation that has a selection coefficient of -1% will take over a population that has 1,000 individuals. So sort of a succinct way of putting this is that natural selection is really good at getting rid of bad mutations. There's probably a fixation of like 10 to the -18. But it's not so good at capturing the good ones. That even a pretty beneficial mutation, say something that makes you 10% fitter, but only have like a 20% chance of actually ending up fixing in the population. That very often even things that are beneficial get lost. So, many of us have this idea in our head that evolution is about optimalism. And in some sense it is, that animals and organisms are adapting to their environment and becoming better tuned to it. It's a really messy, nasty process in that there are lots of things that are beneficial that end up getting lost. But what evolution and natural selection are pretty good, at least in a moderately sized population, getting rid of the bad mutations and not letting them take over. Any questions about these? Yes, sir.
What would you say to the idea of like increasing genetic load, as in like because of our like scientific capabilities, the human population is increasingly like building up, say like mutations that would have otherwise been purged out, but are increasingly becoming commonplace in the human genome?
Yeah, that's a really interesting question. I mean, it's so the question was, what is? Let me paraphrase. You correct me if I'm mistaken. That there's a certain amount of talk in the world about humans have changed our environment. There are things that were strongly deleterious in the past that we can correct for now. Right? A classic example is, let's see, here. At least a third of you are wearing eyeglasses, and probably more of you have contacts. So a big part of that is environmental. Right? It turns out that exposure to bright light as a child, or lack of exposure, predisposes you to becoming nearsighted. But there are genetic contributions. And so, if you were a hunter-gatherer in Africa, not being able to see without glasses would be terrible. If you're a student in America, you fix that pretty easily. It's kind of obnoxious, but it doesn't change your life. So human society is changing the selective environment, and that's something that that's the right way to put it. I don't think that we have a real way of quantifying that. And, I think there is a vein of this thought that is very, what's the right way? Associated with sort of eugenics and, the species just going downhill. We're letting the inferior breed, and that's just terrible, right? That is a terrible line of reasoning. I'm going to teach a whole class about it in spring, about the history of scientific racism and its connections with genetics. It's not on the books yet. You should contact me if you want to take it later. But maybe I didn't. I probably didn't directly answer your question. No, it's all good. But hopefully,
it's a pretty weird.
It's a fraught subject, right? It's something that we have to think about as well. So what does my group do? So my group we do population genetic inference. So inference is a big word. And what that means is that we want to take the data we can observe and learn something about the past from it. And if I had all the data I possibly could, I would love to be able to take a population and sequence every individual in that population for hundreds of generations. And just watch every mutation over time. But typically we can't do that. There are a few very special circumstances where people are starting to do that, but we can't see what happened to that mutation over time. And evolution isn't about a single mutation. If we were to follow some population over time, there are mutations constantly being created as DNA is damaged and passed on to the offspring, and building up a pool of genetic variation in the population. That new mutations are being created, some of them fixed, many of them go extinct, and eventually we end up in some sort of steady state where there's a whole bunch of mutations circulating around. And the data we typically have available to us are snapshot that in 2016 we sequenced 1,000 people around the world. And we know what genetic variation looks like right now. But we want to know about the processes that created that genetic variation. And so my group is focused on building computational tools to help us ask those questions. So given what we observe today, what can we learn about the past, about population sizes and patterns of selection? Any questions?
Can you get data, like 23andMe, they obviously do, like, tons of data sequencing. Do you guys have any way to, like, access that if you gave the person who bought it, like, whatever, yeah, you can use this for like, scientific research, or is that just public or company, like,
Sure. Yeah, interesting question. So 23andMe does not allow access to their database, right? For both ethical reasons and because that is the real value of the company, right? Is that they have genetic data on like 20 million individuals now. It's really interesting. So sorry, there have been bids to buy 23andMe. You can work out from that how much do they think each individual's genome is worth. Something like $1,000. Like your genome is worth more to 23andMe than you pay to have it. But so if you've done 23andMe or something, you can download the raw data and do things with it yourself and potentially contribute to things. I've never been involved in that sort of work, but it does exist. Also something I'll cover in the class on design. So lately in my group, we've been thinking about this thing called the distribution of fitness effects of new mutations, the DFE, which is a big word. And what we think about is a histogram. So on the x-axis, we have the selection coefficient. So 0, remember, means mutation is neutral. -1 would mean that your fitness goes from 1 to 0. So that is something that is meaningful. And the selection coefficient of +1 would mean that individuals that carry that mutation on average have twice as many offspring as individuals who did. So that would be like a super mutation. And so my group has been really interested in trying to understand the shape of this distribution. And it turns out to be important for lots of topics in evolutionary biology, like how genetic diseases are distributed among people, how fast pathogens adapt to new challenges. So we would like to know the shape of this distribution. How likely is it that a mutation is going to be lethal versus neutral versus really, really beneficial? And so I have a question for you now. So let's consider a non-synonymous point mutation. What is non-synonymous? I mean a mutation in a protein-coding gene that changes the amino acid. So we're going to change one amino acid in someone's genome. Qualitatively, what do we think the distribution of selection coefficients is going to look like? Here are some options. So, except for the people in my group, I want you to turn to your neighbor and decide which do you think is the most likely case. And we narrow it down to?
Is it A?
Is it A? How many people thought A? Let's go. How many thought B? So I'll narrow it down. So I'm going to say not B. And one reason I'm going to say not B is the same reason I'm going to say not B. No! And the reason I'm going to say not B or D is that in both these cases, there's a lot of mutations that are greater than 0, that are beneficial. And one thing we've learned from lots of experimental studies and observational studies is that beneficial mutations tend to be pretty rare in most circumstances. And you can think about this as sort of like, this is one of the classic like intelligent design criticisms of evolution, right? Is that we have these highly tailored genetic machines, and most changes we make are going to be bad. Well, it turns out to be true that most mutations are either going to be neutral or reduce the fitness of the organism. So B and D, not so hot. Because of that. That leaves us a few more possibilities. Anyone say C or F? Okay. So this is a little bit harder to eliminate this one. These also tend not to be what we find in experiments. We actually tend to find a bimodal distribution, where there are some mutations that are sort of have modest effects, and some mutations that are really bad. It turns out things tend to look like A or B more. that mutations either tend to have modest effects or they totally screw it up in its research. And between A and E, here's like just sort of empirically we don't see this sort of double hump here. That we do see something more like
What?
anyone choosing?
Why?
Wow. We're all around today. Okay. Well, hopefully this gives you a little bit of insight into what sort of the evolutionary process looks like. There are lots of mutations in the population. Only a small fraction of them are beneficial. Some non-insignificant fraction are very strongly demonstrated as lethal. And then there's sort of a bulk that are sort of neutral or moderately demonstrated. So we have maybe 2 more minutes or so, okay? So I'm going to skip ahead and talk a little bit about, just to highlight some of the work my group is working on. And so this is a project that started many years ago with a group of postdocs and I think 2 universities, both Megan and Alyssa were in work at various points. And we just got published last year. Sort of elaborating this idea of the distribution of fitness effects to think about the case of more than one population. So if we have, say, humans in Africa and humans outside of Africa, we might expect that the selective environment is different. But the same mutation could have different effects on fitness, say, within Africa versus outside of Africa. So that it's not just a single selection coefficient that characterizes that mutation, but at least 2. And so now when we think about a distribution of fitness effects, it's not this one hump distribution, it's actually a 2-dimensional thing, where we think about S1 and S2. And so our question was, what does this look like? And we started fitting models to try and understand, does it look like this? Meaning that S1 and S2 are pretty loosely related, versus S1 and S2 are almost the same, right? Which in sort of roundabout ways can get questions about how mutations, the genome, interacts with the environment, called G by E, versus mutations that interact with each other, called G by G. I think we're also interested in this idea to understand the early stages of how species are formed. And for a more practical point for humans, one of the big issues in doing genome-wide association studies to try and find the gene for X and the gene for Y is that almost all those studies are done in European populations nowadays. And there's a really big question about how much of what we're learning in Europeans will transfer to other populations from them. And so this idea of how related are selection coefficients around the world we think about important implications for understanding how genetic variation in humans is distributed and shaped. And why don't we cut there? I don't think we have time for more diving more into the our particular project, but I'm happy to answer more questions for the next 4 minutes.
That was brilliant.
So what do you guys find in the paper that you published?
What do we find in the paper? So it's much more like this. So we look at humans, Drosophila, and tomatoes. In all cases, it's pretty highly constrained. Humans were actually the least interesting. And then we found very, very few systematic differences in human populations. From an evolutionary perspective, humans are in some sense kind of boring, that we have really low genetic diversity compared to almost any other species you would think of. And part of that is that we're a young species, right? A couple 100,000 years ago, evolved in Africa and has sort of explosively spread around the world. There hasn't been that much time to develop for our genetic diversity to match sort of a huge number of genetics.
Would that also be because of like a complexity thing? Like in my head, correct me if I'm wrong, like genetically, but like in my head, I feel like a human is more complex than like a tomato. It makes sense to me that like there would be less genetic variation because genetic variation would more likely result in like a deleterious mutation, in the sense there's so much complexity and it needs to work so well together. I don't know if that's like completely unfounded or.
Yeah, that's an interesting thought. So I will say plant genomes tend to be much bigger than human genomes. So complexity is a really slippery concept. I think I don't think there's any systematic evidence about humans are easier to break mutations than other species. We tend to think of ourselves as biologically special, but our biology is really shared, right? There's not that much that differentiates a human from any other one, especially at the molecular level.
I do understand. Like, I'm really glad you mentioned it. But I do think it has like some sort of a point though, because like plants can have like multiple set of like chromosomes and still survive. Like a seedless watermelon would have like 3 sets of chromosomes, like which would immediately kill like any other like animal per se. Do you like have an idea of like why certain plants would be able to survive like more like genetic mutations?
So it's really interesting. So I have an active collaboration with Mike Barber, who's in EE here, who is the expert on polyploidy. What you're referring to is polyploidy, that plants and some organisms like salamanders. So y'all have 2 copies of your genome, right? One for your mom, one for your dad. So some plants have 8 or 12. Some salamanders have like 6. And so it turns out that plants and some animals are really good at doubling their genomes or sort of hybridizing and creating new numbers of copies of their genes. And we are actively working to try and understand that. But every time I see Mike ask, so do people know why plants do this so often? No.
There's more.
No.
Or just go back to the slide.
That's your last question. So here's my Twitter handle. Here's my my email is r@arizona.edu. Thank you all.